Welcome to Halteres Presents, a brand-agnostic interview podcast that seeks to objectively highlight the happenings within the world of diagnostics. And now, your hosts, Thayer and Mickey Urdea.
Hello, and welcome to Season 2 of Halteres Presents. My name is Rich Thayer, Managing Partner at Halteres.
And this is Mickey Urdea, Founding Partner at Halteres Associates. Today we have the pleasure of interviewing the author David Quammen, who has written a number of nonfiction books, particularly with regard to infectious diseases, including his book Spillover from 2012, Breathless very recently on SARS-CoV-2, and Chimp in the River, plus Ebola. And we're going to touch on all of the books that I just mentioned in our interview with David today. I got to tell you, he's my favorite science writer. I've read every one of his books, and I just can't wait for you to hear the conversation we had with him.
And now our interview with David Quammen.
So welcome, David. It's a pleasure to have you with us, and we can't wait to talk with you. To start out, please tell us a bit about yourself, including your transition from a Rhodes Scholar who wrote 4 novels to a writer of 18 nonfiction books about science.
Well, thank you, Mickey and Rich. It's good to be with you both. I'm glad for the chance to talk with you. I started out as a fiction writer, yes, as you have said, a long time ago. I published my first novel in 1970, right as I finished my undergraduate degree. And then, yes, went to Oxford on a Rhodes Scholarship for a couple of years, continued writing fiction. But I discovered after the first book how lucky I had been to have a story and to find a good publisher for that story after that first book. It was tricky to do the second one. So I paid my dues between the first book and the second rather than the more conventional way. A young writer pays dues before first book. So I went 13 years between the first book and the second. In the course of that time, I did write some more fiction, but I also started reading and writing nonfiction, mostly about the natural world, which had always been an interest for me. I had always been interested in nature as a kid, and I'd always been interested in writing. And then I happened to have some very superb and influential English teachers, and I had good science teachers, but never a biology teacher that inspired me and mentored me the same way. So I turned into an English major as an undergraduate with a fascination for the novels of William Faulkner, and then started writing fiction myself, published that first novel that I mentioned, and gradually circled back around to my interest in the natural world and in biology. After I moved here to Montana in 73, I started reading nonfiction and saw that some of it was very artful. And I thought, well, that's the sort of thing that I think I might like to write. I started reading J.B.S. Haldane and Annie Dillard and early John McPhee and Stephen Jay Gould and Robert Ardrey and a lot of other things, you know, the great philosophers, Herodotus, Thucydides, historians and philosophers, Diderot. I read a lot about the Enlightenment and decided that I liked the idea of being a nonfiction writer. I could explore the world. I could learn about other people's jobs, other people's thinking, other people's work, and particularly in the realm of field biology, ecology, evolutionary biology, and conservation biology. So my second career began as I started to write about those things, and that eventually led to sort of a third phase, which is when, instead of writing about the ecology and evolutionary biology of big animals in the Amazon or the Congo, I found myself writing about the ecology and evolutionary biology of scary viruses. And 20 years later, I seem still to be in that phase.
Yes. Well, and thank you for staying there.
It's a fascinating as well as an urgently important field, as you well know.
It is. It is. In fact, that's one of the reasons we're so excited to have you on the show.
Sometimes people ask me, well, who's your biggest literary influence as a science writer? And the answer is William Faulkner. The most complex books that I have managed to write, long, carefully, intricately, organically structured books, I hope at least, not obviously structured, not orderly books, but intricately structured books such as The Song of the Dodo, The Tangled Tree, Spillover, Breathless. I could not have written those books. I could not have structured those books if I hadn't immersed myself for years in the novels of William Faulkner. And I even mentioned Faulkner and Absalom on the last page or two of Breathless, this book about the coronavirus, because one of the things I learned besides technique from Faulkner was the slightly more philosophical, epistemological truth that truth itself can never really be fully known from a single point of view, and that the way to assemble truth in its wholeness is to look through diverse points of view. The points of view in Faulkner of different characters, very different characters, telling the same story, and the reader eventually comes to understand the three-dimensional truth of that story by listening to different characters tell that story. Likewise, it seemed to me as I was coming to the end of Breathless that we won't understand this horrible, dramatic experience of COVID-19 and even the mystery of what the origin of the SARS-CoV-2 virus is unless we listen to a lot of diverse, different voices. And I refer to that at the end of Breathless as a literary uncertainty principle, an uncertainty principle that was very important to me. This one didn't come from Werner Heisenberg. It came from William Faulkner.
Interesting.
That's wonderful. Thanks so much.
You must have built that same concept into the Chimp in the River chapter in Spillover with the Voyager, and that point of view of how HIV might have spread and why, and concerns and not. Just fascinating way to have delivered that information.
Thank you, Rich. That 110 pages or so, one of the reasons that my publisher and I eventually pulled it out and turned it into a little self-standing book with a little bit of further writing and introducing was because I think that 110 pages is the most important 110 pages I've ever written.
Wow.
And it was risky in a thrilling way to do what I did by creating sort of a hypothetical character, not fictionalizing fact. I believe that nonfiction should be nonfiction. A quote should be sacred, just like a number in a scientific database is sacred. You don't change those things unless you tell the reader, okay, now I'm going to go into the hypothetical mode and I'm going to create a hypothetical character who's going to do certain things that might have happened that will illuminate what could have happened based on the very solid particulate evidence that we do have. So I feel very strongly about the Chimp in the River. So thank you for for mentioning it.
You've been investigating this journey for more than a decade concerning zoonotic diseases and outbreaks.
It was 23 years ago or so when I started writing about Ebola, and that was because I was on an assignment for National Geographic to do a series of stories about a fellow who was walking across the great remaining pristine forests of Central Africa, from the northeastern corner of the Republic of Congo across Gabon to the Atlantic Ocean, bushwhacking the whole way, following a zigzag compass route to stay as deeply in the forest and as far away from villages and roads as he possibly could to see what was there. And so I walked with him for a series of 4 sections amounting to about 53 days. And we walked in river sandals and river shorts, wading through swamps and across blackwater streams and camping at night on the ground. And he had a crew of African men who were helping him build simple camps, carry the gear, cut a path through the forest with a machete. And I and a photographer were tagging along to do these stories for National Geographic. And at one point, we walked for 10 days through Ebola habitat in northeastern Gabon. And we knew it was Ebola habitat because there had been human outbreaks just a few years earlier in the villages on the edge of this forest block along the upper Ivindo River. And so we knew Ebola was there somewhere in a reservoir host, some animal in which it lived when it wasn't infecting and killing humans. Of course, the reservoir host of Ebola was not known then. It's still not known now, although some people think it's known, but it's not known with certainty now. So it was there in the forest, Ebola, somewhere. And that focuses the mind, as the saying goes, to know that there is an animal somewhere around you that's carrying this deadly virus. and you don't want to interact with it if you can help it. And so I read up on that before and after I did that stretch of walking with him, and I got interested in the virus, and I realized that the question of the reservoir host of Ebola virus, where does it live, and how does it get into humans when an outbreak begins, I realized that's ecology and evolutionary biology. The ecology and evolutionary biology of an invisible creature, a scary virus. And as soon as I realized that, I felt, okay, this is in my comfort zone. This is in my wheelhouse. I can write about viral emergence because I know my way around ecology and evolutionary biology. So that's where this phase of my journey began. It was in that forest called the Minkebi forest block in northeastern Gabon. Me and Mike Fay and a few other men, and Ebola virus.
It ties into your theme of island biogeography as well, doesn't it?
Well, it does in the sense that it's a matter of biological diversity and where it comes from, how it evolves, and where it goes when it disappears and when humans disrupt habitat. And the emergence of new viruses, new zoonotic viruses into humans, that is, viruses that come from wild animals and spill over into humans, that happens in the course of habitat destruction and disruptive interaction between humans and other animals. For instance, capturing them to be exported as live food or killing them to be eaten right away. Bushmeat, we call it. We call it bushmeat when Africans do it, and we call it game when people in Montana and Idaho do it, but it's the same thing. That's where biological diversity is harmed, is lost, and that's where new viruses emerge into the human population. So I think of this all as three great problems that face us on planet Earth, three problems that are all of our own causing. And I think of those as three great brown roiling rivers traveling parallel with some channels linking them. And those three great brown rivers of woe are the loss of biological diversity, the emergence of pandemic threats, particularly in the form of new viruses, and climate change, all running parallel. Some people think that climate change is the problem and it encompasses everything else, loss of biological diversity, emerging diseases, etc. But that's just wrong. And I politely like to emphasize that it's wrong. Climate change is not the ultimate problem that encompasses these. It's a parallel problem. And some emerging diseases are caused by climate change, and some loss of biological diversity is caused by climate change. But in both cases, not all of it. And you have to think about those three things as distinct but interrelated problems of equal magnitude.
That makes sense. I mean, we see diseases that are moving to new parts of the world because of climate change, but that is just a piece of the puzzle.
That's right. Yeah. Some indeed are. Dengue virus fever is moving because mosquitoes are able to expand their ranges farther north and farther south as habitats warm. Malaria likewise is expanding. Tick-borne fevers are expanding, again, because those arthropods, those mosquitoes, those other biting flies, and those ticks are able to expand their ranges out of the tropics and the subtropics into temperate zones, and they bring these diseases with them. Yeah. But there are other diseases that are emerging diseases that are not nearly so clearly linked to climate change. And COVID-19 is probably one of those.
Right. So you made it very clear in your 2012 book, Spillover, that after your interviews with so many people and trying to understand the phenomenon of spillover, that it was likely just a matter of time before the next pandemic was going to occur.
Yes.
And that coronavirus was one of the short list culprits that were likely to participate. What was it like to be in a position where now, after SARS-CoV-2 appears, that what you had communicated so clearly in 2012 actually occurred?
You're right that I did say that in Spillover. Toward the end of my research for the book Spillover, which is published in 2012, I asked a number of the world's leading scientists who study emerging diseases, particularly emerging viruses. I asked them, is there a next big one coming, a next big pandemic coming? And if so, what does it look like? And they told me, Don Burke, a scientist at University of Pittsburgh, and a number of others told me in composite form, what they told me was, yes, there is a pandemic coming. It's bound to happen. We can't say when, but we can make a few calculated guesses about it. It's going to be caused by a virus. It's going to be caused by a virus that comes out of a wild animal, a zoonotic virus. It's going to be caused by a particular kind of virus, a single-stranded RNA virus, and not a DNA virus. Why? Because single-stranded RNA viruses evolve more quickly. They mutate more rapidly and therefore evolve and adapt more quickly to new hosts. What kind of an RNA virus? Well, very possibly an influenza virus, a measles-like virus, or a coronavirus. Where is it going to come from? Well, a wild animal. What kind of wild animal? Well, very possibly a bat. Where might that happen? Well, very possibly at a wet market somewhere. For instance, one of the big wet markets in China. That's all in the book.
Yeah.
But not because I was prescient, because I was listening to the right scientists, and they knew these things were the likelihoods. And so then when this pandemic began in January of 2020, I started getting a lot of calls. One of the first calls I got, it was actually an email, was from the New York Times op-ed page, an op-ed editor at the New York Times. This was in mid-January of 2020, and she said, "Hey, Quammen, you do op-eds for us occasionally. Time for another one. Have you got anything on your mind? Do an op-ed of some sort for us about something that seems to you interesting and important. Oh, for instance, this new virus thing in Wuhan." This was about, January 15th of 2020. And I said, "You bet, I'll do an op-ed for you about this virus in Wuhan." So I wrote and published this op-ed in the New York Times. I think it ran on January 28th. And it said, this could be the big one. This virus in Wuhan could be a pandemic, could kill a lot of people. And that because of that, and because that sort of led people back to my book Spillover from 2012, or some people remembered the book from 2012, I started getting a lot of calls to be a talking head, to be one of the people who were trying to explain this pandemic from early on, January of 2020, to the general public on TV and podcasts, on radio in China. I was in Australia. I was in Tasmania at the time. In Tasmania, in Australia, they were asking me to explain this. Russian TV, US TV and public radio, etc. And some people said, "What does it feel like to be Nostradamus? What does it feel like to have called this, to have predicted this?" And I always made clear that I did not foresee this. I was not prescient. I listened to the right scientists. But the other thing I said to them was, well, I would rather have been wrong. But I was not wrong because these scientists that I had been talking to were not wrong.
Yes, you were very, very clear about the fact that you were communicating from others. So when you look back now, after all this has happened, what could we have done differently? What could we have done to be more prepared?
Well, there's a long list. We could have a long conversation about that. And it begins with paying attention to the fact that human disruptive contact with wild animals exposes us to the great diversity of viruses that live in wild animals. It begins with recognizing that we live in a world of viruses. Viruses are probably the largest single category of biological diversity on this planet, of genomic diversity, viruses. Every species of animal, every species of plant, every kind of bacterium or archaean have their own unique viruses. And most species have many more than one unique virus, lots of viral diversity. Sometimes it's shared among a number of species, but there is Nobody knows how many hundreds of thousands of viruses there are out there just in mammals and birds. And mammals and birds are the animals from whom we get the viruses that infect us. Mammals, that's obvious because we are a mammal. So the viruses that infect mammals are more likely to be able to adapt a little bit and infect humans. We also get viruses from birds. All of the influenzas come from wild aquatic birds. For some reason, those influenza viruses that infect birds also have the capacity to infect humans. And sometimes they come through pigs, and influenzas are also quite well adapted to pigs. And the viruses that infect pigs uniquely sometimes swap sections with the bird viruses that infect pigs, and out comes a virus that's extraordinarily dangerous to humans. Influenza viruses have this trick where they can swap whole sections of their genome from one to another if they're infecting the same animal at the same time. And coronaviruses have a similar trick that they do in a slightly different way. So, back to your question, what could we have done differently? Well, years before this pandemic began, we could have started recognizing that we endanger ourselves and other people by all the disruptive contact that we have with wild animals and the ecosystems in which they live. That's the first thing. And then there's a whole list of other things. If you accept the reality that we're disrupting a lot of wild animals in their ecosystems, there are still other things that you can do to prevent viruses that spill over into one human or into two humans from turning into big outbreaks that become epidemics, that become pandemics. Smart surveillance of human and animal creatures at the interfaces where humans and animals interact is one of those things. But there's a whole list of specifics that can adumbrate what I mean by smart surveillance.
David, those are very sobering comments about the world in which we live today. Turning it about a little bit, now that you've had the chance to study the scientific response to COVID-19 and all of the 95 plus interviews that you did for Breathless, what are some of the great achievements and who are some of the key heroes that you uncovered?
Thanks, Rich. There are great achievements and there are heroes, known heroes and unknown heroes. But with the achievements, I would say the list begins with very quickly sequencing the genome of this virus. The genome is 30,000 letters of RNA long. Sequencing that genome from a sample that came from a human patient and making that sequence public, putting it up on the web as soon as it was assembled. And there was a team of scientists who did that. They were led by Yongzhen Zhang in Shanghai and assisted by a great partner of his, a molecular evolutionary biologist named Eddie Holmes, the great Edward C. Holmes, but everybody in the virology world knows him as Eddie, at the University of Sydney. He's British, but he's based at the University of Sydney. And the two of them and their colleagues publicized the genome early in the morning of January 11th, 2020, Sydney and Shanghai time. And they put it up on a website run out of Edinburgh, where it was the middle of the night, January 10th to 11th. And at that point, people such as Tony Fauci and Barney Graham of the Vaccine Research Center in Washington and Bethesda and many others were waiting for that sequence. Tony Fauci had told Barney Graham, look, you just be ready to start work on a vaccine using the mRNA vaccine platform concept. And as soon as we get the genome, be ready to go, and I will get you the money, I will get you the support, and you can start work on that vaccine. So Tony Fauci sees the genome come up on this Edinburgh website about 8 o'clock on a Friday night, January 10th, 2020 in Washington. Barney Graham sees it come up. Immediately, Barney is calling his young colleagues and saying, get ready, we're going to go on with this genome, we're going to build an mRNA vaccine. Let's go to the mattresses and get this done. And I think it was 65 days later, the first prototype of that vaccine, the Moderna vaccine, went into the arm of the first volunteer, and that was an extraordinary achievement that saved a lot of lives. Meanwhile, other people were working equally quickly on vaccines of other types in other places. The Oxford AstraZeneca vaccine was being made very quickly in England, and the Pfizer vaccine was being made very quickly, and other vaccines in other countries were being produced very quickly. Those are some of the heroes of this, and that was one of the great achievements. Not the only one, but one of the most conspicuous ones. And it wasn't just Tony Fauci and Barney Graham and Eddie Holmes and Yongzhen Zhang. It was a lot of young people, postdocs and graduate students and lab assistants and people we've never heard of. But they were all working at top speed, and that's one of the reasons my book is titled Breathless.
That's great. By the way, Eddie Holmes is an old friend of mine. We published 4 papers together. I've known him for 30 years. He's a great guy.
There we go. The great and you know then, Mickey, that in the world of virology, if anybody says, well, that may be true, but Eddie says this, you don't have to explain who Eddie is.
That's absolutely true. The one and only. So you pointed out in Spillover that molecular testing techniques used to identify suspected viral pathogens are only part of the answer regarding what could trigger the next outbreak. What's probably equally important is to understand the entire public health ecosystem involved, and in particular the epidemiology associated with the viral infection. So, what are some of the techniques and approaches that are being used to aid this field work? And for instance, is there merit in broad surveillance efforts in wild animal populations to predict the next spillover event, perhaps using eDNA or other emerging screening tools?
Well, that's a really good and complicated question, as you well know, Mickey. How do we use science to identify, predict, or at least detect the next big one before it gets big, in order perhaps to prevent it from going big? And there are 2 schools of thought on this. They're not mutually exclusive, but they tend to be divisive, and that is the viral discovery approach and the surveillance and response approach. And you know what I'm talking about, but I'll explain this for your listeners very simply. There is a project called the Global Virome Project, conceived by a fellow named Dennis Carroll, very smart guy. And the concept is, let's go out and sample animals all over the world and look for the viruses that they contain and create a great database, a great encyclopedia of the viruses that exist in mammals. And then let's look at them and try and predict which of them are most dangerous as potential human pandemic pathogens, and then take steps to prevent those pandemics from happening, keeping people away from those particular viruses by keeping them away from those particular animals. That's sort of the most broad, capacious approach to this. And a different school of thought says, no, we can't afford to try and learn about every virus in every animal any more than we can count the grains of sand on a seashore. We need to look carefully for where people are stepping on starfish and stingrays and sharp conch shells on that beach. We need to look for the interactions that yield damaging results. We need to do smart surveillance by, for instance, doing continuous serological sampling, blood sampling of people who work at the interface between wildlife, domestic animals, and people, and the animals in those places. Smart surveillance, proactive blood sampling of people who work in chicken farms, massive poultry operations. Proactive blood sampling of people who live partly on wildlife, on bushmeat, whatever you want to call it. Proactive blood sampling of the animals that are carried into markets to be sold as food. So that's a more targeted approach. The smart surveillance, active surveillance particularly through blood serological sampling, looking for antibodies, using other forms of testing to detect viruses that not viruses that might spill over, but viruses that already have shown signs of spilling over into humans. Maybe they haven't caused any disease yet. Maybe they haven't made more than a single person sick, but let's find those situations and then be ready to contain those situations. So both of these approaches have their partisans and have their merits, and it would be nice to be able to do both. But of course, always the pie is finite and can only be cut into so many slices. The pie of public commitment, the pie of political capital, and most obviously the pie of money. Those pies are finite, and some people, including Eddie Holmes, argue that you got to make decisions. You can't have your pie and eat it too. Now I'm mixing metaphors with cakes, of course. Eddie and some of his colleagues have published on this subject, saying, no, we can't just do broad scale, comprehensive viral discovery. We have to do targeted surveillance.
Yeah.
And that's a very interesting segue into the next question. Coming right in the heels of the COVID-19 pandemic, and not well covered yet in the general media, we appear to be in the midst of another highly pathogenic avian influenza outbreak, impacting the California condors and the raptors and the waterfowl, geese, ducks, and everything else. Quite significant. Tens of millions of animals have been culled or have died already from that just this year. What are some of the things that our audience might be thinking about in terms of what they may be able to do to help stem the impact of this outbreak or the opportunity for spillover to continue into the next year or so?
Yeah, that's important, Rich, and that's the little cloud on the distant horizon, no larger than a fist, that we have to look at very carefully because it's coming toward us, and it might carry more than its share of thunder and lightning. This is something, as I guess you know, that I published an op-ed in the New York Times on this about a week ago, on the subject of high pathogenicity avian influenza, the latest version of H5N1 influenza, which is circulating among wild birds all over the planet and killing a lot of wild birds, including individuals of some endangered species. You mentioned the California condor, and there are others, endangered species of birds that are being hit by this high path avian influenza, but also domestic birds, poultry, in massive industrial scale poultry operations. There have been millions of domestic birds culled because they were sick or because they might get sick or because they might spread the virus in the last few years. And occasionally this avian influenza spills over into mammals and infects mammals. And there's a long list of mammals that it has infected: some dolphins, some porpoises, some sea lions, some mink. There's a list, and I won't go farther because my memory won't be exactly accurate. But it has gotten into quite a number of mammals, foxes also, I believe. And that, of course, shows that it's capable of replicating in a mammal, making a mammal sick, killing a mammal. And that's a warning signal to us that we could be next. This H5N1 has gotten into some people too, and the latest version of H5N1 doesn't seem to be as virulent, as pathogenic in people as some earlier versions of H5N1, but it has that capacity. It goes back and forth. As you know, influenzas evolve quickly.
Mm-hmm.
And so it is very possible that this could turn back into not just an influenza that is transmissible to the occasional human, and not just one that kills most of the humans that it infects, 53%, I think, in some of the earlier versions of H5N1, but it could also take that next step and be not just capable of infecting humans, not just capable of killing humans, but very capable of transmitting from one human to another. And it's mutations are random, but Darwinian natural selection is always there to shape those mutations in a way that allows a highly adaptive creature to seize new opportunities. And we don't want ourselves to be that next opportunity. So, back to your question of what can we be doing, what should we be doing? Among other things, as I said in the op-ed in the New York Times, people should think about the fact that we now have 33 billion chickens on this planet at a given moment. Most of those in large industrial scale poultry operations. Birds are highly concentrated, living in dense aggregations where they can be infected by wild birds carrying the virus. And once they get it, they can transmit it like wildfire through a big population of domestic poultry. And when that happens, that domestic poultry operation is functioning as a great petri dish, offering opportunity for this virus to evolve in a way that could infect humans. You've got hundreds of thousands of chickens out there. Who knows how many of them at a given moment are infected with this new virus? And you've got people walking among them. And what is the virus doing? The virus is replicating in thousands of different birds potentially. And when it's doing that, millions and millions and millions of replications per day and constantly mutating. It's an invitation to Darwinian evolution by natural selection of a virus that could infect and transmit among humans. So we need to think about the fact that we as consumers are creating that situation. 33 billion chickens at a given moment on this planet. And somebody informed me, a fellow emailed me after my op-ed was published, and he said, yeah, 33 billion chickens in a given moment, but there's turnover. So we go through something like 70 billion chickens, 70 billion, in the course of a year. Boy, are we giving avian influenzas a great opportunity to evolve every which way.
Yeah. Is there any evidence to date that there's mammal-to-mammal transmission?
There is a little bit of inference in that regard, Mickey. I don't think there is solid, concrete molecular evidence, but there is epidemiological evidence. There are places where there have been considerable die-offs of mammals. I think it was sea lions in Chile. There was a great die-off, and excuse me if my memory is wrong about the right species and country, but I think that's right.
Mm-hmm.
So we have sea lions in Chile. If they have been transmitting this virus from individual to individual, that would account for the patterns that we've seen. We don't, as far as I know, have the molecular evidence for that, but it is ominous. It is very concerning to see any large mammalian die-off that might be attributable to this virus because it suggests mammal-to-mammal transmission.
Yeah. Seen some recent indications that the notion of vaccinating chickens is becoming more and more realistic. They, of course, do this in China now, but the U.S. has not been willing to do that. But perhaps that's where we're going to go. What do you think of that?
Well, I think that it's an important idea, and I don't know what the constraints may be on it. I haven't researched that particular thing, but yes, absolutely. I mean, when I was researching spillover, I spent time in a border area of Cambodia near the Vietnamese border with a wildlife veterinarian named Billy Karish, great wildlife veterinarian who then was with the Wildlife Conservation Society. And I went with him. That was an earlier occasion when bird flu was a great concern. So we went chicken vaccinating in villages up there along the Vietnamese border of Cambodia, dressed in, full PPE. We went into these villages at night and started saying, may we vaccinate your chickens? They probably thought we were from Mars, but Billy patiently explained that it was for the good of the chickens and for the good of the people. We were vaccinating against Newcastle disease, which is a very contagious poultry disease, but also doing some sort of prototype work to vaccinate against avian influenza. So that has been done and can be done, and I'm sure should be done. But we need to very quickly develop a poultry vaccine against this latest strain of H5N1.
Right.
David, you've mentioned a couple of times during this interview bats. And the potential role bats may play as a host for various viruses. And there have, of course, been many reports concerning the role of bats and the countless species of viruses that they may act as hosts for around the world, including suspicion that they may act as a host for Ebola, though that hasn't yet been proven.
Right.
Just what is it about bats that enables them to serve as the host for so many different viruses?
That is a fascinating and important mystery. Why bats? And as you know, Rich, I devote an entire chapter to that in Spillover, the question, why bats? So I asked a lot of scientists the same question you just asked me, why bats? And there are some complicated but interrelated answers. First of all, it does at least seem that bats are disproportionately represented as reservoir hosts of a lot of new viruses and old viruses that are dangerous to humans, that spill over into humans. Hendra virus comes from bats in Australia. Marburg virus comes from bats in Africa. Ebola virus is suspected to come from bats, but we don't have the gold standard proof yet. Other viruses, Australian bat Lissa virus, rabies virus itself, Australian bat Lissa virus is related to rabies. A number of others, Nipah virus in Malaysia and Bangladesh. The original SARS virus from horseshoe bats in southern China, and most likely SARS-CoV-2, but that hasn't yet been proven. So bats, bats, bats. Yes, there is this pattern of bats. They seem to be overly represented as the reservoir host. Why might that be? Well, for one reason, bats are an extraordinarily diverse order of mammals. There are roughly 1,400 species of bats, and that means that 1 in every 5 species of mammal on planet Earth is a species of bat. This extraordinary diversity. The only order that's more speciose, more abundant in different species, is rodents, and rodents also are implicated as reservoir hosts in many emerging viruses. So with bats, it's that, but there's something else that distinguishes bats from rodents, and that is that they fly. They're the only truly flying mammal, and the fact that they have achieved flight has occurred with the necessity of some trade-offs in their evolutionary history and in their physiology. For a mammal to fly generates great physiological stress on its body, and there is a hypothesis and work done to investigate that hypothesis that the physiological stress that bats undergo to fly releases free radicals, naked reactive molecules in their bodies that is potentially provocative to their immune systems. There is a concern, there is a suspicion that their immune systems would react, if they were as sensitive as the immune systems in most mammals, to these free radicals, these suspect elements afloat in their bloodstreams, that their immune systems would react and they would suffer autoimmune disease, inflammation against their own bodies, against the biochemistry of their own bodies, if their immune systems were as sensitively attuned as the immune systems of most mammals. So the hypothesis is that over the 50 million years or so of bat evolution, their immune systems have been downregulated to be less sensitive toward antigens of various different sorts, suspicious molecules afloat in their blood. And that downregulating of their immune systems over millions of years perhaps makes it more likely that other alien elements, such as viruses, can exist in their bodies without being immediately attacked and cleared by their immune systems, and that they may tolerate low-level infections more readily than other kinds of mammals, so that they might be serving as reservoir hosts, carrying populations of viruses that are replicating at relatively low levels and causing little or no inflammation, and not getting into battles with their immune system for that reason.
Fascinating.
Some of the most recent work I've seen very interesting is that they seem to have high levels of interferon at any given time, even without an infection,
Mhm.
or they're poised to secrete a large amount of interferon very quickly after an infection, which is very interesting and rather different from other animals as well.
Yes, yeah, yep. And I'm going to leave that on your authority, Mickey.
Okay.
By the way, David, this is just fascinating.
Yeah.
Good.
I'm
Thanks so
a
much.
more recent convert to your writing, but I'm telling you, this is enthralling. Thank
Well,
you so much.
you're welcome.
Yeah.
Very welcome, Rich. This
Yeah.
is always endlessly fascinating stuff to talk about. I'm at work on another magazine piece right now for the New York Times Magazine on the question of the origins of the SARS-2 virus, that whole controversy that's ongoing, and the question of how the general public understands that controversy. There's a lot of, well, I don't want to talk too much about it because the piece isn't written yet. But so I've been talking with Eddie Holmes again, and with Christian Anderson, and with scientists all over the world on various different sides of the mystery of the origins of the coronavirus. Some people say, oh, it's a zoonotic virus. It's a natural spillover. There's a lot of evidence in support of that. It's not final. And there are other people who are persistently arguing that, oh, it came from a lab leak. Look at this coincidence. Look at that coincidence. A different kind of argument, not so empirical. It's more forensic. And meanwhile, the general public is saying, what should we believe? What should we believe? Anyway, I don't want to say much more about that. But the
That's
point I was going
fair
to
enough.
make was that here I am back on that subject again. I've been writing about it in this case for 3 years and more, and in the general sense about emerging viruses and their ecology and evolution for about 25 years. And it's still just fascinating to me. And I learn new things every day.
Yeah.
Yeah.
David, we did a project for World Health Organization a year ago where they asked us to look at the opportunities for improved pandemic preparedness. And surveillance is one thing that comes up. It's frequently not funded, though there are sequencing banks and networks set up all over the world for those reasons, maybe including eDNA and not just other more traditional
Yeah.
sources for sequencing, etc.
Instead of actually taking it from an organism, you take it from a place like the air, soil, water, some place where it's been deposited for one reason or another. And then if you can figure out how it got there, being able to use that as information about the presence of specific organisms over time. So ecologists love it in particular. They'll, for instance, take a stab of soil and look at and sequence the layers and determine how changes in organisms that were in or around that area changed over time.
Yeah, yeah. Well, that brings a couple of things very, very strongly and vividly to my mind. The most immediate is, of course, the environmental sampling of RNA in the Wannan seafood wholesale market, from which the metagenomic sequences have just been published finally after 3 years by China. But even before that, it puts me back in memory of the pioneering work of Norman Pace in his association with Carl Woese when he developed methods after Carl Woese had been developing very, very laborious but revealing methods of genome sequencing in the mid 1970s to try and figure out what is the shape of the tree of life. And then his young friend Norman came along and said, let's do this with samples drawn from hot springs in Yellowstone Park. Let's do metagenomic sequencing using these techniques just from a bucket of slime that we pull out of Oyster Springs in Yellowstone National Park and see what we find. And look, Carl, we found more of your new kingdom of life. We found more archaeans living in slime in the hot pots, the thermal features of Yellowstone National Park. And so there is this great explosion of discoveries thanks to Norman, God bless his soul, who I've been privileged to interview multiple times, and Carl Woese, who I never met. But I wrote a book about this that you may or may not be aware of called The Tangled Tree that was published back in 2018.
Yeah, I love it.
And so those guys were doing eDNA, right?
Essentially, they were. It's probably redefinition. Norm is an old friend too. I haven't seen him in a very long time. But your depiction of that whole situation with Archaea and Carl and it's just so well done. I just enjoyed that book so very
Well,
much.
thank you very much. This is why my publishers are saintly in their patience. I moved to Simon and Schuster in about 2014 from a different publisher, 2014 or 2015, and they said, "What do you want to do? What do you want to write your next book on, David? We'll take it, whatever it is." And I told them, "Well, I've got 3 ideas that I'm very interested in. One is cancer as an evolutionary phenomenon. Another is prion diseases. And the third is reshaping the tree of life through discoveries involving horizontal gene transfer." And they said, "Oh, we'll take the one about the tree of life." And God help them and God help me. They didn't realize what they were getting us in for, and that what I was really going to have to write to deal with that subject was a history of molecular phylogenetics. But I never said to them, "I'm writing a history of molecular phylogenetics." I said, "I'm writing a book about the reshaping of the tree of life and this amazing phenomenon called horizontal gene transfer and this amazing guy named Carl Woese."
Right. Yeah. Prions would be a wonderful one. I was actually a postdoc at UC San Francisco when Stan Prusner's lab was doing all the preliminary work on prions.
Well, it is fascinating to me, yeah. And Stan Prusner and I have been in touch, um,
Yeah,
and he has not been discouraging of that idea.
I'll bet.
uh, but I haven't gotten there yet.
You convey it so well. You're such a wonderful
Exactly.
storyteller.
Thank
It's
you.
Exactly.
it's a great pleasure.
Thank you very
Uh,
much. Thank you.
so you have a new book that was just published, Heartbeat of the Wild, which you were kind enough to share with us, and we greatly enjoyed it. It's a wonderful set of your travel pieces that originally appeared in National Geographic. Please share with our listeners a bit about your thinking about the wild, which is clearly a major theme there, island biogeography, or other themes from the book.
Well, you're certainly right that there's a lot of travel as well as a lot of science that went into these pieces. I don't think of them as travel pieces because I was always simply going on field excursions, expeditions with scientists. It was work every step of the way, although there was a lot of adventure, a lot of enjoyment going on. Essentially, it's 20 or 21 pieces from National Geographic over the course of 20 years in which I was assigned to write about wild creatures in wild places and the conservation of biological diversity, what works and what doesn't in these different situations around the world. So National Geographic would say to me, for most of this time, I was a contributing writer for National Geographic, which meant that I was not an employee, I was on a contract to write 3 feature stories every year for them. And that could be my suggestion or their suggestion. And usually it was their suggestion because I found that it was easier to let them persuade themselves that they wanted a story on something and then persuade me rather than me trying to persuade them. And it was always interesting things. They would say to me, "David, do you want to do the Serengeti lion with Nick Nichols, a great photographer that I worked with on a lot of these stories? Do you want to do the Serengeti lion with Nick?" Well, of course I want to do the Serengeti lion with Nick. Do you want to do elephants of northern Kenya with Nick? Of course I want to do elephants of northern Kenya. Do you want to go to the Arctic on a ship for 6 weeks with an exploration, a party of explorers who are sampling biological diversity in Franz Josef Land, the Arctic islands of Russia? Yes, I want to go there for 6 weeks on a boat to the Russian Arctic. Actually, with that one, I hesitated slightly because 6 weeks is a long time, and I'm happily married, and usually I try to be gone for no longer than a month. But my wife, she's also an author, and she's a very adventurous soul. And so before I said yes to the Arctic trip, I ran it by her. Should I go on this expedition exploring biological diversity in the Russian Arctic for 6 weeks. And she said, well, first of all, yes, you should do it because it will be interesting and important. And secondly, if they had asked me, I would have already said yes. So I went on that trip, and I went to Mozambique, to Gorongosa National Park, and I went to the Russian Far East, to the Kamchatka Peninsula, to write a story about efforts to conserve salmon in the great rivers of the Kamchatka Peninsula. I went to a number of these places and wrote stories about them. And National Geographic Books wanted me to pull these stories together into a book, but we all agreed that this should be more than just a collection of my previously published articles, that it should be interconnected in a more organic and significant and frankly artful way, if possible. So I chose these pieces. They were all about wild places, wild creatures, and conservation. And I put them together into this book, and I did more reporting. I updated the situations on all of them. I wrote connective material that had to do with the editorial process, the human relationships, the later stories of what happened in these different places, connective material to turn it into a book, and then a foreword and an afterword exploring the concept of what do we mean by wild? What is wild? And what I say in the foreword is that, wildness is very hard to define, but what we mean by it generally is something that is biological, that involves ecosystems that are large and significant and connected and intact enough to create creatures and relationships that have this living and breathing diversity, unpredictability, connectedness, wildness, ferocity in some cases. And it's different from anything else that you might call wild. It's different from a black hole. It's different from an erupting volcano. It's different from the great spot on the planet Jupiter. It is biological wildness, and that requires size, the size of an ecosystem, the sheer scope of an ecosystem, connectivity, processes that link it in a way that conserves this great scale of biological diversity with every level of creature, from big predators that sometimes kill and eat humans and routinely kill and eat wildebeest and water buffalo and the big prey animals, down to the organisms that can only be detected with microbiological methods, the viruses, the bacteria, the archaea, the whole thing. When you have that all intact and sizable enough, functioning with all of the processes, predation, herbivory, scavenging, decomposition, photosynthesis, that characterize a fully intact ecosystem, then that's wildness. And then it has that heartbeat that I refer to in the title.
It's a wonderful book, and it was really very interesting to read your follow-ups at the end of each of the original stories. Yeah,
Thank you.
that's really interesting.
And the book, I'll just add, the book is dedicated to Nick Nichols and Mike Fay, dedicated to this photographer that I worked with on so many of these stories, who originally brought me into National Geographic because we wanted to work together, and Mike Fay, the explorer that led that expedition across Congo and Gabon that I mentioned earlier, where among the many things I discovered what it was like to walk through Ebola habitat.
Amazing. Are you going to get a chance to return to the cancer book you were working on before you were interrupted by COVID-19?
I hope so. Yeah.
Yeah.
As I've already mentioned, that was one of my three books on the list, was when Simon & Schuster brought me into their corral.
Yeah.
So I have been interested in that subject, cancer as an evolutionary phenomenon, for a long time, at least 15 years. About 15 years ago, I published a cover story in the magazine Harper's about this. It was called Contagious Cancer, and it was about the fact that cancer evolves, a tumor evolves just the way an organism evolves by Darwinian natural selection. I can explain that if you want, but I won't go into it right at this second. And that is a very important perspective on cancer, both for understanding how it works, what happens within a given individual who is afflicted with a tumor, how that tumor changes, what that tumor does. For pure understanding, it's important, but also for very practical matters of treatment, oncology. Understanding cancer as an evolutionary phenomenon is a school of thought that is growing in importance in clinical oncology. And so, because of its intellectual fascinations and also because of its health implications, I do want to get back to that book project.
Yeah, and I understand you were in Tasmania, specifically look at the devil's facial tumor story.
That's
Is that
right.
correct?
Yeah, this famous case that people perhaps have heard of, the Tasmanian devil, this charming, unique species of marsupial omnivore that survives only on the island of Tasmania, is afflicted with a genuinely transmissible tumor, a facial tumor. And this affliction is called devil facial tumor disease. It began in about 1996, and it has spread through Tasmanian devils all across the island, Tasmania being the southernmost state of Australia, the one that's separate as an island below mainland Australia. And this tumor has been rampaging through the Tasmanian devil population. Some people say, "Oh, that means it's a virus, right? It's a contagious virus that triggers cancer." No, it is known clearly with very, very rock solid hard molecular evidence that this is a contagious tumor that passes from one individual to another. It's supposed to be canonical that cancer is not an infectious disease, but it's not canonical. It's contingent on certain circumstances, and when there are other circumstances, cancer can do this. And that's the lesson of the Tasmanian devil. And that's why I was in Tasmania in February of 2020 when the COVID pandemic exploded, and I was spending half of my time traipsing through the bush with Tasmanian devil biologists, watching them trap these animals and take samples of them, and in some cases cull them or ban them. And I was spending the other half of my time in the Australian Broadcasting Corporation studios doing radio interviews about this pandemic coronavirus. It was a weird month for me.
I'll bet. Is it correct? Do I remember correctly that it now appears that some Tasmanian devils are resistant to the cancer?
Yes, it does appear. It does appear that the cancer has been sweeping across the island from the northeastern corner toward the southwest. The southwestern corner is the wildest and most remote habitat for the devil, and it does seem, the latest I've heard, that there is some evidence that the devils in the southwestern corner may have some sort of a natural resistance to the virus, which would be very good news for the population, for the existence of the species. There's also there's evidence that there has been a second tumor, a second independent contagious cancer that has arisen in the devil population, is not yet as transmissible as the first one, but has also managed to do some spreading from one individual devil to another. It's just not as aggressive and as transmissible as the first one so far, but it seems to be an independent lineage based on molecular evidence.
Fascinating.
David, thank you so much for your time and the fascinating discussion.
It's been a joy to speak with you and to hear from you. We know our listeners are going to love it, and it was more than we hoped for, and we hoped for a lot. So thank you.
Thank you both very much. I really appreciate the depth of your interest and the depth of this conversation. I've really enjoyed it.
Wonderful.
That's great.
Halteres Presents is produced by Halteres Associates, a US-based bioscience consultancy that provides strategic and tactical services in the areas of diagnostics, medical devices, and life science research to clients of all sizes. The views, information, or opinions expressed expressed during the episode are solely those of the individuals involved, and Halteres Associates is not responsible for any errors or omissions, or for the results obtained from the use of this information. The information provided in this episode is for informational or educational purposes only, and is not intended or implied to be a substitute for professional medical advice, diagnosis, or treatment. Halteres Associates would like to say thank you to this episode's guest or guests, and thank you for listening to this episode of Halteres Presents.